Okay, so welcome back, everybody. So it's really with pleasure that I introduce the second and last speaker of today, that is Luca Rossini. So Luca has been a junior fellows of BrIAS in the last 2 years. He is a postdoc here at ULB with a Marie Curie scholarship. And he has a quite interesting background that since I see in his preview of the slides, he will speak about it. I don't want to spoil. So I will let him the stage and let's see the show.
So thank you for the introduction, and actually thank you, David, for introducing many concepts that I'm going to recall in my presentation, because we have seen that plants should be protected, and so David showed us just one face of the coin, that is fungi, pathogenic fungi. So now I'm talking about insects, so about what insect science, called also entomology, but with a different approach. So the importance of models in plant protection against insect pest. And so, as David said before, so the same disclaimer of interdisciplinary approach will be also the same of my presentation, because this is basically my background. So I have a bachelor in physics, but then so I get passionate with the world of agronomy, and with my PhD I put everything together, because ecological modeling and applied entomology. And now, so since October 2023, I am a Marie Curie researcher at ULB in the Department of Control Engineering and System Analysis. So my Marie Curie project is called Pest Finder, and our goal is just combining different disciplines to deal with the problem of controlling insect pest population in a farm. So basically applying all the techniques that are very well known in control engineering, but it's not just control engineering, as you will see, is insect science, is mathematics, is metrology, just with the final goal. So in this seminar you will see how over the years we arrived to formulate this project and what we are doing now. But I mean, as you said, as you saw in my first slide, I'm a hybrid person, and being hybrid is not always easy, because you have to interact with people with different backgrounds. So in my everyday life I interact with engineers, entomologists, applied mathematicians, metrologists, and everyone has his own language. So basically we are a kind of translator, so hybrid people act as a kind of translator. And we are also source of a lot of jokes, because, I mean, I heard many definitions of myself, because it's quite hard to explain who I am. Probably I don't know who I am. And, but just to give you an idea, according to the engineers, I am an insect guy who plays with numbers. But on the other way around, according to the entomologist, I am a physicist with 6 legs. So if you don't understand the jokes of 6 legs, just wait 10 minutes and you will know why. And so it's quite hard to understand what is an insect guy who plays with numbers or a physicist with 6 legs. So I thought that since one of the theme of BrIAS of this year is AI, why don't we ask to AI to explain what are those 2 definitions? And according to ChatGPT, this is the result. So it is something quite weird, but so according to AI, it is what I should be. But jokes apart, my main topic of research is rational pest control. And because now in smart agriculture, we want to we want to reduce the amount of inputs in cultivated fields, because every time we use an input, we are somehow modifying, so leading the sustainability of the environment, and we are also increasing the production cost. So basically, we should provide the inputs in cultivated fields only where and when they are really needed. And to do that, and in the particular case of insect pest, insect science alone is not enough. So we should combine different scientific fields together to look at this problem in a different way. And so again, besides insect science, we need applied mathematics, because we can, if we have models to provide some forecast, we can know where and when we should provide the inputs in the cultivated fields. But we need also metrology, because we need to collect some data and some information from the field, because in this way we can combine everything together through control engineering and automation. And so in this seminar, I'm trying to explain how all those fields of research are connected in between each other. And I will start introducing the problem. So basically, agronomy and insect science, and then I will move towards the second topic, that is applied mathematics, because I will explain which are the common techniques that we use to mathematically describe insect populations. We will move towards metrology. So I'll show you which are the complexities of collecting data in insect science and so in entomology. And then I will combine everything together with control engineering. So let's start from the first step. What is IPM, Integrated Pest Management? And the core question of this first part of the seminar is, so what is a model-based integrated pest management? So basically, why we need models? And if we have to describe what is, in an ideal world, integrated pest management, we can use a scheme like that. So we have a farm as a central point, because for a farmer, all the farming process is the core business. So a farm is like a company, and they should have some income. But from the farm, we can get some information through the monitoring. And monitoring can be, for instance, pest abundance, the infection level in the case of diseases, can be measuring the environmental parameters. So all information that we can get from the field. Those information can be somehow processed by models, and models can simulate different scenarios. Those different scenarios will be helpful to take decisions, and the decision is choosing the best control action, to reduce the level, the abundance of the pest into the field in a more sustainable way. So basically, we should start in IPM with the lowest impact for the environment control strategy, and then we should we should increase the level if needed. The control action is going back to the field, but there are other two dashed lines in this scheme, because it's true that the information that we get from the field can be the input for the models, but models can somehow, and this is the role of control engineering, somehow suggest where and when we should collect the data. And this is something that, at least in insect science, is quite new. Again, if we include the control action, so the effect of the control action, into models, we can simulate different scenarios, and those different scenarios can provide more realistic, so we can provide a more faithful situation of the future situation of the field, such that the decision can be taken on more realistic, on something that is closer to the reality. But unfortunately, this is still a kind of ideal world, because So if you look at the normal management of farms, this is the real scheme. So basically you have an approximate monitoring, so a few information that comes from the field, and based on those few information, the core model is not an equation, but is the experience of farmers, because they are so experienced in sometimes that they can process the information, and they can find the best control strategy. But I mean, it is something that is still, so for the level that we want to reach now with precision agriculture is something that is not anymore sufficient. And this is the reason why we should improve the scheme, developing models and new techniques to move towards what we call decision support systems. And so now, this is our main goal. And smart agriculture, from the pest control point of view, is solving this problem. And this is the reason why we need all the different, so the interaction of all the different fields of research that we have seen at the beginning. But now, so we are moving towards the second part of the presentation, that is mathematical models. So from insect science, we are moving towards applied mathematics. And the core problem here is to formulate reliable models with a biological meaning. So biological meaning is the most important part, because we don't want just a mathematical game that more or less reproduce the trend of the data that we can collect from the field, but we want that the biology is suggesting us the assumption to build the models. So, because, I mean, we don't want to lose the connection with something that we can observe and measure. And since the biology is the main component of the formulation of this problem, let's start from scratch, introducing what is an insect. And if we want to describe in a very simple way what is an insect, we can say that is an animal with 6 legs. So just now you understand the joke that I made at the beginning. But from the modeling point of view, there are few features that we can account, and the most important one is that insects are ectoderms. It means that the duration of the life cycle depends on the external environment, mainly depends on temperature. So at low or high temperature, we will have longer life cycles. And then at the optimal temperature, so for all the species, we can always identify an optimal temperature where the life cycle is the shortest one. And this is one of the main features that we should account if we want to build a reliable model. And then there is another feature that is really important, that the life cycle of insects is divided in stages. Those stages are clearly identifiable. So if you ask to an entomologist, can you explain how many stages compose the life cycle of this species, you will get an exact answer, because all the stages are morphologically identified. Then we have more or less 1.5 million of species, and we will have very different and complex, less complex or more complex life cycles. And not all the species have the division in males and females, because there are some cases where there is a phenomenon called parthenogenesis, where females are just reproducing females for some species. So the sex division is something that is not always happen. And now, this is just the first point, but this is a hint for all the people involved in image recognition or computer vision. If you want to recognize an insect, you have to keep in mind that there are, in all the species that we have different sizes, so from a fraction of a millimeter up to 20 centimeters. and different shapes. But those different shapes are, so we can have different shapes between the different species, but also different shapes in between the different stages of the same species. So an egg, the shape of an egg is different from the shape of an adult. So you should keep in mind the stage that you are targeting and that you want to recognize. And insects are everywhere. So if you think that it's just a problem of natural environments or of agricultural fields, this is not the case. So insects are, of course, in natural environment, of course, in cultivated fields, but they are everywhere because they can infest greenhouses, they can infest warehouses, we can infest our houses because there is a branch of entomology that is just facing problems in urban environments. Think about all the vector-borne diseases. It's like malaria, Zika, this kind of viruses. They are all vector-borne diseases. So it's a mosquito, the vector. So basically, again, is insect science. But now, more or less, we know what is an insect. Let's see how can we obtain more or less a good model to describe mathematically an insect population. So this is something that I reached more or less towards the end of my PhD. It was during my first year of postdoc, because analyzing the literature, what you see, the old literature, what you see is always a single model to describe a single species. But actually, if you look at the difference in between, and the analogies between the different life cycles, so from the biological point of view, and the equations, so the mathematical point of view, there are a lot of common points. It means that we can, so generally speaking, we can put all the mechanisms that we can find all together in a single scheme, but when we know, for instance, how many stages compose the life cycle, or we know the mechanisms of reproduction, or for instance, if we have which is the sex division, so if we have males and females, or if we don't have the male, we can obtain the particularized model. So this, which is the philosophy behind this scheme? So each compartment represents a single life stage. So a life stage that if you ask to an entomologist, can you define what is an egg? So basically, from the mathematical point of view, is this compartment. And so this compartment is described by an equation, and the solution of this equation is the number of individuals over time. This number of individuals over time and in all the stages basically is regulated by some transition rates. Those transition rates are development rate, if we are meaning the transition from the stage to the next one, can be a mortality rate if the individuals are living the life cycle, can be a reproduction rate if we have a production of new individuals coming into the first one. And so we can also have the sex ratio, because so the sex ratio is the ratio between males and females, and is not always 50-50 in some populations. The extreme case of parthenogenesis, the sex ratio is 1. It means that this term is disappearing. So this chain of compartments is a single line up to the last preimaginal stage, so the last young stage, and the division happens only when we have the adult. Why? Because it makes sense dividing the population in the adult, just because it's the only stage that is devoted to reproduction. So it doesn't make sense to twice the number of equations for something that we cannot observe. Then, in the female stage, we can have different mechanisms. Again, different species, different mechanisms. We can have, for instance, a division in between non-mated females and mated females. It means that the beta 2 transition rate is set to 0. So we are canceling out this arrow here. Or we can have different fertility rates based on the aging of the females. So it's what we call gonotrophic cycles, that is typical of some species of mosquito. So as you can see here, we include all the possible mechanisms, but if you don't ask to an entomologist to define the life cycle and the main mechanisms, you cannot apply this scheme to a particular case. And this is what we mean that we need biological assumptions. From a mathematical point of view, this scheme is just represented by a system of ordinary differential equations, and the key role is played by the transition rates, because those functions, generally speaking, contain, so the parameters of those functions can be estimated through laboratory experiments. So those experiments are quite complex, but so we can obtain some parameters, and they connect the mechanisms that we have in the population with the external environment. So the Trojan horse for the environmental parameters is inside the transition rates. This system can be nonlinear. It's not necessarily linear, because if you have some dependencies, for instance, on the abundance, so the population abundance of some states, you can use the transition rates to include this dependency. So this was just the first step that we carried out, so we were quite surprised, because at the beginning we applied this model in different case of studies, and we were surprised to see that at least we were able to reproduce the trend of the data, and there is no correction from the field data. It's just an open loop simulation. But again, so precision agriculture is providing inputs where and when. So from an ideal point of view, we could just answer to the question when we should apply the treatments. We are not answering to the second question, that is where. And a first attempt of spatialization so just comes from analyzing the mathematical structure and the stability of the system. And as a result, you can obtain a risk index. This risk index is parameterized, contains all the transition rates, and connects, again, the environment, so the environment and the parameters that identify the species. But this is something that still is not practical, because it's not considering one of the most important parts that we need. So basically, we need to know how insects move in between the different areas of our field. This is the reason why, again, we went back to entomology, to insect science, and we started to analyze which are the most common approach that are used from insects to move. So, as you can imagine, there are a lot of different mechanisms, and so whatever is the species, you can always identify which is the mobile stage. So the stage that can cover long distances. We can have, for instance, passive movement. So there are stages, some stages that is like a powder is carried by the wind or by water. In other cases, you might have an active flight, so an adult with wings. But anyhow, you can always identify which is the stage that is forced to stay in the plant. It's like an egg, so an egg cannot move, while an adult with wings can, so is devoted to spread the outbreak in other places. And I mean, if you identify those features, the game to spatialize the model is to use what we call a meta-population approach. So you can divide your field. Can be a single field, can be a bigger area, but so the concept is exactly the same. And in a series of, in a grid, and you obtain some subparcels. So each subparcel can be described by the system of equations that we saw in the previous slides. But now we have some migration rates that appear only in the equation that is identifying the mobile stage. So two parcels can exchange individuals if and only if they share one boundary. This is the strategy that we use to spatialize the model. And this was a good step in the theory, but up to now we are still looking for some good data for validating the model. And as you will see, collecting the data is a big problem in our field. But now, we could answer to where and when, but still we have a problem, because so once we were just looking at some data collected in a climatic chamber, when everything is controlled. You know how many eggs you had at the beginning, you know exactly which is the temperature, the diet, all the things that you cannot control in a natural environment. And this is the situation that you that you see from the data set. Basically, you can track the population over time in all the stages, and you know exactly which is the initial condition. So, but as you can see here, the peaks of the population in the different stages, they are not overlapped, but there is a time shift. So this time shift is something that is recalled in the biology. So if the development is something that is related to a series of biochemical reactions, even if the environmental conditions are the optimal ones, we cannot go, so we cannot decrease this time that we have to wait. So this scenario was quite mind-blowing at the beginning, because we said, okay, we want to test the model, because usually in a field, in an open field, you just track a single stage, but you don't know anything about the other stages. In a climatic chamber, you can track all the stages. So this is the perfect bench test to validate a model. But if you look at the simulations here, so this is not the simulation of this data set, it's just a plot to show you the behavior of the simulation. All the peaks of the different stages are overlapped. So we don't have this time shift. So again, we made a little step back, and we started from the beginning. And we said, what is missing? Is missing an important concept, that is the minimum development time. So if we place 100 eggs in a climatic chamber, and those eggs are laid in the same moment, at the same moment, so the minimum development time is the time that we have to wait to observe at least one egg that is hatching, or a transition to the next stage for the other life stages. So from the mathematical point of view, this led us to revise a bit the system, and from a system of ordinary differential equations, we went to a system of delayed differential equations. Let's say that this is more or less the philosophy behind our modeling approach. And as you can see here, so this is a simulation just to show you the behavior of the model. Now you have the time shift. So it means that the concept of minimum development time is something that is really important if we want to improve simulations. But now there is a common belief in many, so talking about with entomologists, there is the common belief that if we have a model and this model is validated, this model is ready to be used to be included in a decision support system. So basically, if we can describe a series of experimental data, we can use the model for predictions. And now we have this question: are these models ready for predictions? The answer can be yes, but if and only if you know which are your initial conditions in the field. So basically, you should estimate in a clear way when the outbreak, so when is the time zero of the outbreak, and the initial abundance of the population. This is it is something that, trust me, you cannot know easily. So basically, we are moving towards control engineering. But before doing that, what can be the solution? Probably we need a feedback from the field. So now we are moving, before jumping towards control engineering, we need an intermediate step through metrology. Because if you think that the data collection process in insect science is something that is easy, the answer is no. And now let's try to answer to this question. Let's try to understand what is a measurement in insect science, and then we are trying to find a solution to combine the models and the few measurements that we can get. So, what is a measurement in insect science? And there are different techniques, and I will show you the pros and cons of the different techniques. I want to introduce the first one that actually is one of the most common, that is the visual inspection. Visual inspection basically is defining a protocol and a certain number of plants that you want to inspect, and a stage that you want to target. And just to give you an example, so let's consider a vineyard. And this vineyard is composed of 2,000 plants per hectare. And we want to track the population of one of the main pests of vineyards, at least in some areas of Europe, that is Lobesia botrana. And you can see why it's an important pest. So basically, it's causing, besides the direct damage, is also opening the door to a lot of molds. So basically, what we call secondary host in this case. So your protocol can be inspecting 5 bunch of grape per plants, and if you are super expert, you can, each measurement is just taking 30 seconds. But this is just for one single point, and if I'm not bad in math, you need 80 hours per week, because you don't want just a single point, you want a time series. So you should repeat this operation for all the growing season, that usually is 6 months, 5 months, it depends on the crop. But if you want to track the population all the year, you should repeat this operation all the year. So it is something that is extremely time consuming. And this is still a kind of lucky case, because the height of a grape plant is something like that. So something that you can easily reach. But imagine that you want to track the population of this guy, Tomaiella parvicornis. So this guy is 2 millimeters long, and is basically a huge pest of stone pines. So it's an alien species for stone pines, and it's a huge problem in Rome. So I was involved in some research, and I can say that the damage is something that is really, really huge. But the living environment is the canopy of a stone pine. It means that if you want to take some samples, you need to use a crane. So basically you have to rent a crane, you need someone with a special permission that is going up, is taking samples, you have to go to the lab, count the specimen on the different samples, and you repeat this operation on different plants and all the weeks. Another technique is quite curious, that is called frappage, or branch shaking. So I used this video from a colleague, Nicolò Di Sora, and you will see this guy many times in this in this presentation. And so it consists of having a white sheet on the ground, and you just shake the branch of the plant, and you are going to count how many individuals are falling on the sheet. So you understand that you should repeat this operation for a sufficiently high number of plants in your field to have a good statistics. But again, it's just for one single point. So you have to go there every week and you have to repeat the operation. But so likely enough, we have some good friends that are traps. So traps is a kind of way to automatize the data collection process in entomology. But again, the process is very easy to understand. So there is a lure, and this lure usually is reproducing the smell of food or the sexual attraction. So it basically is reproducing something, some biological aspect of the target species. So with the lure, we are just attracting the individuals, and then there is a catching part. So usually it's a sticky band like this, or is a bottle. And so on the sticky band or inside the bottle, we can count how many individuals that we had, so that we caught. And but every time you have to inspect one trap, you have to do this game. So basically, you go there, and again we can see Nicolò doing the job, counting how many individuals are on the sticky band, taking notes, and doing this operation for all the traps that we have deployed in a field. So this is something that automatizes the process, but still there is one common problem to all the techniques that we saw. So we don't know which is the proportion of the individuals that we are counting and the actual population into the field. So at the point that recently we had a bit of brainstorming about that, and so we raised a question. So what is measuring a trap? And this is actually an open question, and is an open door for all the metrologists. So please help us, because it is something that we still don't understand. If you think that just counting insects is something easy, you're wrong, because so the identification of the species is something that is extremely difficult, and there is a branch of insect science that is called taxonomy that is just defining all the morphological and genetic traits to classify the different species. And so usually so insects are classified based on the morphological traits. It's not just looking at the color or a general shape, but it's something that can be, so it's really analyzing the shape of a leg, the shape of the wings, the shape of the antennae. So, and in this way you can define which is the species. Just to conclude the part related to measurements, so in all the cases that we have seen, we are just counting directly the individuals. But there are some cases where this is not possible, because we can only see the damage. So this is a case of it's not an insect, it's a mite, Phytoptus avellane, but it's a mite pest of hazelnut plantation. So what you can see is the damage that is called pseudo-gall or big bud. So basically so this guy is going to infest the healthy buds, and the plant as a response is producing the big buds, so the pseudo-gall. But actually, if you dissect one of those galls, you can see that you have a colony inside, and this is really something small, something really small. And if you want to spend some time at the stereo microscope to count the individuals, this is the scenario that you see. So basically, all those small points here are the mites. Again, here the only way is just counting the galls and trying to estimate the infestation level. So it's true that measurements in entomology is something that really needs help, but those information can be somehow used and connected with measurements, so with models. And the answer is yes. And So we are moving towards control engineering, because the first time I interacted with Emanuele, I was always trying to look for a solution to estimate the initial conditions, to switch from the description to the prediction. And so the answer was, yes, but it's our job. Okay, so we started the interaction, and so basically the idea is to revise the model and to build a model that is reproducing the measurement process. And the first attempt that we did to solve the problem of the initial conditions was applying one of the most common techniques in control engineering, that is the Kalman filter. And basically is a two-step algorithm. I know that there are control engineers here, so please don't blame me. And but so just to explain in simple words, is starting from the assumption that a model is something wrong by definition, because it's just a schematic description of a natural phenomenon. On the other way around, measurements are affected by an uncertainty, but the Kalman filter is trying to combine those two information to obtain an improved simulation. And so we applied this scheme, so this framework, to the case of Drosophila suzuki. That is exactly the case that we saw at the beginning in the first validation of the ODE model. And so we compared two scenarios. The first scenario is so randomly generating some initial condition and solving the model. And as you can see, the dependency on the initial condition is quite hard. So it is quite high. So there is a huge variability. And we cannot, of course, we cannot rely on a simulation like that to take decisions. But if we include the feedback from the data, we are just we are reducing the dependency on the initial conditions, and we are improving somehow the trend of the simulation. So the trend of the simulation is closer to the field data. So this was our first attempt of applying control engineering to insect science, and but then this opened a lot of problems that are really interesting, because now we are moving towards the last part of the presentation, that is more or less what we are doing now. So putting everything together. So we have models, we have some measurements, and now we know that we can combine everything together through control engineering. And the problem one of the problems that we are facing now is the optimal sensing problem. Because one of the first questions that I always heard when in insect science, so entomologists are starting a monitoring survey, is how many traps I should use and where I should place them, because this is one of the most recurring questions. So what we are trying to do now is to use the properties of the Kalman filter to solve what is called an optimal sensing problem. So we want to define, given a number of traps that we can afford or that we can manage, so should we leave them in the same place, or can we move them to improve the information that we get from the field? So basically, this is what we are trying to do. So it's a model-driven data collection. So asking to the model where we should put the trap to the next step. Then another. So this is the last slide. Another problem that we are facing is it's a farming problem. So there is a common belief that usually the pest population should be the lowest one into the field, but actually this is not the truth, because so the main goal of a farm is to maximize the profit. And I mean, if we keep the population low into the field and we use control, so treatments, so this is just increasing the production cost, and sometimes it's not worthy, so related to the yield that we can gain. So basically, we should build a model that is taking into account the plant as well, so the yield, and so the model now is going to be a kind of predator-prey model, where the insect pest is the predator, our fruits are the prey. So based on that model, we can formulate an optimization problem, and we may wonder when is the best time. So given a number of treatments that we can afford, and sometimes it's something that is fixed by the law, so we cannot go beyond a certain number of treatments for a given pest in a given crop. So the first question is, we need 3 treatments, if 3 treatments are allowed. The second question is, when is the best timing? And this is something that we can, so it's a question that we can answer through an optimization problem. So this is another ongoing work. So just to conclude, as you can see, so as you saw in this seminar, insect science cannot be left alone, and it's just combining and looking at the problem from different point of view that we can have very good solution to the problems. And with this I have concluded, and I open for questions, and thank you for your attention.
So, thank you, Luca. Are there questions?
Thanks for the presentation, Luca. So it's again from a profane point of view and neophyte in the field. So by approaching, let's say, pest control and pest monitoring, there's always sort of a question that arises. So it's clear from what your presentation that it's very hard to do pest monitoring with the, let's say, with the visual inspections and so on. It's quite, an abnormal amount of work. But having some devices that, check for you, you will go for the single pest monitoring or the disease that the pest causes. So do you also integrate this kind of, let's say, in your model, so this kind of, inspection, so to see if the pest is also virulent state? Because we always, associate the presence of the pest okay, the presence is enough to tell you that it's causing the disease, but you should also check or not the presence of the disease.
Okay. So you mean with pests that are carrying diseases, or
no,
it's just
pest
damage?
Damages.
Okay. No, because so with the pest vectors, yeah, this is this is something that is extremely complicated. But yeah, so this is one of the main problem actually, because you should relate. So in classical integrated pest management, so there is the concept of economic threshold. So it's a certain level of the population that I should not overcome, because after that threshold, I will start to lose money. But this is something that is quite empirical. So the idea, so what you say is true, yeah, because so a high pest abundance, we always do the assumption that if the population is high, the risk is high, so basically we will have damages. But yeah, so we should look at the damage as well. So, but the damage is something that is indirect, because it's really hard to correlate the pest abundance that you have 3 problems. The first one is the connection in between what you observe, what you count, and the actual population into the field. The second problem is connecting the damage with the actual population. The third problem is connecting everything together. So for some species, let's say that the damage, so they consider the threshold as the number of fruit per plant damaged. That is something that is related not on the pest abundance, on the population level, but is based on something that you can observe. But yeah, so in my opinion, that should be interconnected. But the problem is, how can we connect them? So this is the big open question.
There was a question there.
Thank you. My question is, considering the fact that we can also have multiple pests that can be of infestation in a field, and you also have multiple environmental factors, you have different growth stages of the pest. Is it possible to get a model that can cater for all these challenges that plant may face through pest infestation?
Okay, so in the case where you have multiple pests that are infesting the same plant,
Yes.
is that the question? Okay,
Yes.
yeah, of course you can. Because the general approach that we tried, so that we formulated, is something that can be, so that can describe the life cycle. Now, so besides the optimization problems and optimal sensing problems, what we are trying to improve is the different trophic levels. So connecting the different trophic levels, because one case can be when you have this is a kind of double predator-prey model, because you have two predators that are, and the prey is the same, that is the plant. But in other cases, you might have three trophic level, so plant, pest, and natural enemy. And this is something that is extremely interesting, because when I talk about control strategies, I don't mean just only agrochemicals.
Okay.
It can be whatever
Okay.
control strategy. So entomopathogenic fungi, predators, parasitoids, and of course connecting multi-trophic levels is something that is really interesting. And yeah, we can do it.
Oh.
We can do it. Yeah. With the same approach. Yeah.
There is another question.
Thank you for your talk, Luca. You showed us a patchwork of plots with various levels of populations, and you made the assumption that only the plots which had a boundary with another plot would have the pest communicating.
Yeah.
But of course, we have also situations where the situation is much more blurred because the pests are extremely mobile. In this case, we can have a patchwork of damage which does not translate in the same patchwork of pest abundance. And how is it possible for, what you call that, control engineering to solve this type of issue?
Actually, it's a question for you, probably. No. I will try to give my point of view. Yeah, so it's true that, as we said at the beginning, so when we were talking about the transition rates, so in between the different stages, so the development, the mortality, the philosophy behind is that this is a kind of Trojan horse that to connect the biology and the population dynamics that we can observe. So the transition rates in our spatialized models more or less is the same. But while the biological knowledge on the thermal biology, so basically how the temperature or other environmental factors affect the development and the mortality and the other transition rates is quite well known, at least for some species of agricultural interest. So the spatial patterns is something that depends on measurements first. So basically it's a question mark. The second fact is that we don't know exactly all the mechanisms of migration. So it is something that should be explored a bit more, because then the challenge is estimating the migration rates. This is the main point.
Maybe if I can complement, as a matter of fact, so the model is with some migration that is possible and some other that is not. Of course, if I know that a species can at most do 200 meters per day because they walk, you can have a graph of how these things evolve that depends on that. And then you have a lot of surprises. For instance, lately we received one of the very few specialized data that we have ever seen. And there were some patchwork that we could not understand until we spoke with the person that collected the data. And she told us, yes, this is in a greenhouse. And we discovered that there were these mites that were walking on the structure of the greenhouse. So absolutely, I mean, the mechanisms there are much more complex than what we can measure in a lab.
Yes, I mean, there is something that, while for the description of the life cycle, we can be more or less general enough. So, and then we can go to the specific case. Here, I think we should face case by case, because then you have also the cases. So, depends on the scale. So a field scale, a greenhouse scale, and this is a first context. Then you can have some long migrations where you have some individuals that are carried by winds or current, and it's quite common for a lot of species. So your initial outbreak depends on the meteorological factor, but at the macro scale. So yeah, I would say that it's quite unexplored and interesting to explore.
But there is one thing to be said, that, for instance, for geographical movements, the thermal aspects can be interesting to understand when there are bridges between geographical areas that usually don't communicate. So if I have a mountain range that I have to pass through, maybe it's a summer that is particularly hot, the summer in which something goes on the other side. So there are these aspects that anyway come back from the biology.
Million of years of coevolution. So yeah.
Any other question? I might know. I'm joking.
Oh, you can.
Okay, so I would thank again Luca for his talk.
Thank you.